Okay, good morning or noon. Good noon. We'll go with noon. Hello, everyone. Today I have a few things for you here. Hey, guys. Thanks for saying hello. I appreciate that. So, you might know that I did finish grading the exams, but I have not posted them yet. So I'm going to do that pretty much immediately, soon after our class today. Certainly by the end of the business day, so to speak. You'll have your exams graded. I will probably try to get your current participation score updated as well, so that when I post to Moodle, those will both be there. Um, your homework 3, I was trying to get done by today. Things haven't gone as well as I would have liked. I prepared a lecture last night for this morning for my grad class, and it has completely disappeared. I saved the PowerPoint. I made a new folder to put it in. Everything was set, and it's nowhere to be found. I think OneDrive has something funny going on. I may be able to find it eventually, but it has been a morning. For the final exam, I got a question here. The final exam, I believe that's correct. It should be essentially whatever LSU says it should be. I'll just go ahead and double check that with you here. Our syllabus, what does our syllabus say? Syllabus currently says April 29th, but that's the wrong class. Don't believe that one. That's the other class. Sorry about that. Okay, what I do at this time of the semester is I always come through and I double check what's on the syllabus, which I'm doing right now with you. Tuesday, April 27th, 5:30 to 7:30. Then the other thing I'll do is I'll just search for it. LSU final exam schedule spring. And then ours is a standard class period. We meet on Tuesday, Thursday, and we meet at noon. So that should be yes. So I would say yes, that is correct. That is what I'm expecting. We'll talk a little bit more about it on Thursday, but answer to your question for now is yes. Okay, you see the grades posted here as a kind of how the class did. Overall, there were more B's than anything, some more C's than A's, and certainly more were C or less than, or I guess the dividing line is probably something like an 80. And in fact, the average for this exam, let's see, let me just do this one thing real quick. The average for the exam was a 76. So not as easy as the first exam, but there were a fair amount of you that did pretty well. Whenever there are 10 plus people that are below D or below, is a bit concerning. And I do start to think whether or not I need a curve. But just as kind of a quick reminder, exam number 1, I can show the screen if I move it correctly. Exam number 1 was a little bit of an opposite curve, right? We had exam 1 was very skewed to the right, exam 2 skewed a bit to the left. Between the two, I think, is probably what's typical. So if they were both like exam 2, I'd probably be a little more inclined to go ahead and curve it, the second one, right away. For now, I'm not going to curve. I did obviously apply lots of partial credit um, while grading. Um, so that's just the current status. My goal is still to have your homework 3 assignment graded and back to you by Thursday. Then we'll go through homework 4 on Thursday in class. Okay, so with that, what I want to talk to you about today is kind of a special topic, sort of a deal, and this is membrane bioreactors. So we previously talked about membranes, and we've also talked about activated sludge systems, and we have also, fix up the fish can for you, we've also dealt with kind of the growth processes for bacteria in activated sludge systems. And membrane bioreactors are going to give us an advantage in a few ways. So if you think back, and by the way, I'm not going to test you specifically on, we're not covering any new quantitative tools today. We're not covering new equations or something like that. We're going to review some past things, so I think it's probably worthwhile. And I could ask you a membrane bioreactor type of question because you know about membranes, because you know about bioreactors. Those are certainly both within the scope of what I could ask you on the final. And then, maybe the challenge for you would be to keep separate the particular types of equations that you need and things like that. But I would certainly make that straightforward. I'll talk more about the exam next time, but I wanted to preface this to kind of tell you what to expect today. I'm going to include a few research highlights that have some relevant related research in this topic area that I think you might find interesting. So that's my plan today, to share that with you, and then we'll really come back and talk about the final on Thursday and go over the homeworks. Okay, so a typical wastewater treatment system, we have some sort of pre-treatment, activated sludge chamber, so probably sedimentation, activated sludge, and then another sedimentation basin. Excuse me. From here, we would recycle some of the sludge to make sure we have lots of bacteria, and then treat whatever the rest of the sludge is. Maybe try to recover some of the nutrients, maybe use it as a landfill, send it to the landfill, or use it as land application as a fertilizer for some crops. In a membrane bioreactor, we have a little bit of a different system. So instead of the sedimentation tank to separate all of the bacteria after growing the bacteria and making them reproduce rapidly, consume the waste products, the waste junk, and make cells and digested waste products, instead of that, we have this membrane bioreactor. So we don't need the sedimentation, because the sedimentation is there to separate the solids from the liquid. And we really just need most of the cells taken out, and then we can treat the effluent here a little further as needed. But the sedimentation basin does a pretty good job. Here we have this membrane, and we often insert it into the bioreactor itself, or maybe sometimes we put it side stream where we have the membrane here, we take water out, and I guess take that through into the next, into the treated effluent. That's where we would take it. So you can operate it side stream, and then whatever is kind of the retentate goes back in. So that would be side stream, or you just insert it into the container itself, and you're just essentially pulling clean water out continuously. And so you end up accumulating sludge on the bottom, and you gather that sludge every so often, maybe pipe it straight out, and do the same sludge processing. So the advantage here is you don't have to worry about that sedimentation. That means you don't have to worry so much about what kind of bacteria are growing, so long as they're consuming oxygen and the organic stuff that's in there and multiplying. And then you don't have to worry too much. It's okay if the sludge floats to an extent. The air also serves to help clean the membranes. So as you've got lots of bubbles happening, we have to physically shake or backwash these membranes pretty frequently to keep them from getting too fouled. So physical action with the bubbles can help with that. There are some extra costs here in that you have to use pressure to, like a vacuum to pull the water or some other pressurization to push water through. However, the advantages are you can end up with higher bacterial concentration. So this gives high X, where X was the mass of microbes per volume. Whereas the activated sludge, that's the standard. Bioreactors have higher X. What that allows us to do, if you think about where in our equations that X matters, it matters in terms of how much substrate will be consumed, and it matters in terms of the rate at which it's going to be consumed, right? So with higher X, with so many bacteria that would normally, that level of bacteria, we would have some escape. We would fill the sedimentation basin too much. It would not be able to handle this much bacteria, and we wouldn't be able to recycle enough to even maintain that. Well, here, the bacteria are never going anywhere. They're just stuck in the system until we decide to withdraw them. So in that case, we can have a much better control on how long the solids, the bacteria stay in there, and we can end up getting more treatment with less space. So more, or perhaps you could say better, treatment with less space. Now, it does come at a bit of a cost, higher energy and higher infrastructure in the first place. Membranes do have a cost that's different than a sedimentation basin, but in some sense, you've gotten rid of the sedimentation basin altogether, and you've been able to shrink the bioreactor itself. So the places you see these most often are in wastewater treatment plants that for some reason have been restricted in land use. So while I was in Michigan, I actually visited a couple, and both were next to a river. And one of them had railroad tracks on one side and a river on the other side, and they had this little plot of land. The city around them grew bigger, and then the wastewater treatment plant had to accommodate for the greater flow and the larger demands, and essentially was having a hard time meeting that demand, and so they needed something a little more innovative to use a smaller space constraint because they couldn't afford to buy more property. They were limited in that way, and so they went with a membrane bioreactor, and it ended up working quite well for them. Okay, so essentially all that stuff that I just mentioned is kind of repeated here. So there is a trade-off, and there's a reason that not every single bioreactor is a membrane bioreactor, and those are these trade-off costs. But essentially it's doing the same thing. You'll see it'll be kind of a sludge type of system. We're aerating it. Same process is happening. We just are using that membrane instead of the sedimentation basin. Okay, so just a couple pictures here about what this would look like. I pulled this from online, and we see here these essentially these racks or these modules of multiple membranes, and these pipes coming here, these are going to be essentially applying negative pressure to pull water through these membrane cassettes. And chances are that these are either flat sheets that are, and you can see these manifolds go from the main line down into each membrane, individual membrane module. And so either these are flat sheets that are just, let me take this one over here, for example, that are just submersed into the water like that. And so then water is being withdrawn from either side, going in and up and out like that or something. Or maybe these are modules of hollow fiber membranes that are similarly suspended from these just in lots and lots of little fibers. So one or the other, this is a depiction of kind of what it might look like in a system in a moment where it's not actively bubbling. Because it'd be hard to see if this was actively bubbling and got a bunch of sludge everywhere and all of that. So essentially that's the equipment setup. Here's another example, and here they are installing flat sheet membranes, and you can see they're all in a nice little row in racks, and so it's just constantly pulling water through, and you'd have a system where it would be bubbling and helping to scour these membranes with tiny bubbles during the operation. So you see in this middle pane, this is filled with what looks to be pretty clean water, and then during operation, you see it's kind of all muddy and foamy and stuff. But each of these lines is, going to the manifold and extracting water out of all of the membranes. Okay, in terms of water quality standards, we talked about this previously, and so just as a quick reminder, we design our systems with that receiving water in mind to meet the BOD requirements, to meet these different requirements. And one of the requirements that we haven't talked a lot about is simply nutrients, so nitrogen and phosphorus in particular. And one of the things that membrane bioreactors do is, given that elevated concentration of bacteria, they can actually remove the nitrogen and phosphorus to a greater extent than a typical activated sludge system. So that's one of the advantages there. This is a review. I took this set of slides from my other course. We didn't really touch on the types of pollutants and all that for wastewater in that other course because it's primarily just physical and chemical processes. So I'm going to skip over this. You already know about this. We've already covered this before. So just as a reminder, I brought this up already, but essentially the reason membrane bioreactors are helpful and they have that advantage, they have that added X. They keep a larger initial concentration of X, so you don't have any sort of lag phase when food comes in, and essentially you are higher up on this plot, and that log of viable cell concentration, you can keep it in a place where they are actively growing and reproducing and consuming lots and lots of substrate, but they are not in that kind of stationary phase. So that's sort of the goal, is to keep a high amount of them and keep them hungry, constantly, hungry enough to be eating as much as they can, and providing a whole bunch of food for them. Again, we've talked about the monod kinetics and everything, and essentially it's that maximum specific growth rate coefficient. We want to be pretty much getting near that, right? So for a given specific growth rate coefficient. The condition where that is almost the same thing as mu is the condition where Ks is very small in comparison to S, right? So theoretically, we'll probably never quite reach that maximum. You know, we'd have to give an infinite amount of food or whatever, but we can get pretty much close. If this goes away, then it's just mu S divided by S. So at least in comparison, that just means that the higher the amount of food we give, the closer we are to this. And so we can give a lot more food if we have a lot more bacteria there. So that food to mass ratio is important, and having simply a higher mass of bacteria means we can handle more food. Okay, again, this is pretty much review here. And essentially we're just basically saying that how that X factors in. It's going to factor in for the substrate utilization rate because that's related to the growth rate. So if we were to put mu X into Rg here, the rate at which we're consuming substrate is negative the growth rate divided by the yield coefficient. So again, all that's to say, you've learned this before, but that's just to say that the more bacteria, the faster they're going to consume substrate, and that's going to benefit our treatment level. Okay, so this is just an elaboration on this. I'm going to skip through it because you've already seen this. Of course, if you have questions, very happy to help answer them. You know, you're working on a homework right now that's dealing with some of these equations, so please let me know. Okay, so one thing I wanted to comment on here is some of the current research that takes a look at conventional versus membrane bioreactor effectiveness. And so highlighting here, this is detection of viruses by qPCR and basically these DNA type techniques, and cell culture in wastewater samples 2008 to 2012. So we have these membrane bioreactor wastewater treatment plants, and we have different samples. We have after the preliminary treatment, after the MBR treatment, after the UV disinfection, and then we have all of those in the same case except after a conventional activated sludge system. So in terms of like pathogen removal for viruses, we actually don't see a huge difference. You know, if we're to take a close look at what the MBR versus the sedimentation essentially is doing, we look at these samples here, compare them to these ones here, and then looking at, given these 11 different treatment plants, different samples, or 8 in this case, we could take a look at how many adenoviruses or PCR enteroviruses or noroviruses we see. And, you know, it's not particularly different here because we don't expect these processes to change it much. Now, I think there are other aspects that are different. If we were to take a look at, let's say, BOD removal for these two different ones, we should see a big difference. And I think that's one thing that I wanted to show, but I didn't quite find the data on that. But typically, I think if a conventional gives you about 95% removal of BOD, an MBR will get like 99, maybe 99.5%, something like that. So a significant amount more removal. Okay, so this brings me to a research topic that I've been involved with for a few years now, where we want to make use of photocatalysis. And I think I may have mentioned photocatalysis a little bit back when we were talking about disinfection technologies. But in the case of a membrane bioreactor, it actually is potentially a good spot to use photocatalysis because, like we were just looking at, a lot of these systems are going to use some sort of an activated sludge system and then an ultraviolet disinfection step afterwards. Now, in the case of the conventional, maybe you'll have chlorine. You could have chlorine after MBRs as well, but the fact that you're able to remove more junk with the membranes means that you are, I guess, more able to apply UV light effectively. So you, first of all, you can remove the larger pathogens. Something like, even though you're not getting viruses, you can get the cryptosporidium, you can get most of the bacteria themselves, and essentially that sets you up better to use post-treatment processes that are affected negatively by dissolved organics and do not do very well against certain kind of pathogens, the larger ones in particular. So the idea with photocatalysis is that maybe you can take the membrane itself and operate it in an inside-out flow regime where you have water coming in, as you see here, and then exiting on the outside here. You could add your photocatalyst particles, these little white stars, shine light on it, especially if you're already doing the UV disinfection, you could just use that light. So you're getting the UV disinfection, but you're also irradiating the photocatalysts, which are then going to produce reactive oxygen species. And so as water is coming through the membrane and coming out the other side, suddenly they meet the UV light and the photocatalyst producing these radicals. And so you can keep the dissolved organics out and away from the system. They are going to pass through and exist and be concentrated in the retentate, stay in the bioreactor itself. And the smaller particles and the viruses that might come through, you have some filtration here, and then the photocatalysts can be destroying them on their way out. And so what that would look like schematically would be you have your activated sludge, a side stream setup where you pull some water out and do your operation here. And normally, you might have the membrane and then you'd have UV. So here we're just proposing to combine these two by keeping the membrane as a side stream instead of immersed, right? We saw some pictures of immersed, and I kind of mentioned you could take it out and do it separate. That's what we're doing here. So that's the concept. And along the way, when we were working on that project, well, first of all, let me show you some of the results, because the group I was a postdoc at Michigan State University back in 2016, and we were doing some work with a membrane that was coated. We took a ceramic membrane, tubular like this, and coated it with TiO2 that had already been done by the time I was there. And then we were checking for photocatalytic inactivation of human adenovirus 40. So it's an adenovirus. It's a known pathogen. And we were checking to see, okay, well, what happens if we use microfiltration only? It turns out that we had some removal of the virus, probably because it would adsorb to the membrane. Maybe some of it just happened to be removed by the membrane. It's not likely, but certainly, some of the pores might be smaller than others and could have some physical removal like that. We also tried with the with the photocatalysts and the membrane, but no ultraviolet light. So the system was in place as it should be, except we didn't shine the light. And then when we shine the light, we see a significant increase in the destruction of viruses. So that's about 1 log difference. So instead of removing 99%, we went to 99.9%. I'm sorry, I think I misspoke. This one in the middle, I think this was with the UV but without the TiO2. So without the photocatalyst, but we did have the ultraviolet light running. Because the ultraviolet light should kill some viruses, but not very quickly. Not as quickly as the water is running through the system. So essentially, at the end of the day, our point was that we did kill some viruses, and we did see that this concept, this technology concept, was working. Okay, so then from that, we realized that one of the problems in our system was that to grow this human adenovirus, HAD40 here, HADV40, we actually had to use a serum type media to make the viruses happy. So in these experiments, it was not just the little virus particles, whatever they happen to look like, you know, maybe like some little alien lander thing or something. Let's give him a face. Mr. Virus Man. Okay, so not just the little virus guy, but also lots of organic matter in solution that potentially stuck to the photocatalyst surface. So if we have a photocatalyst surface creating these reactive oxygen, if it reacts here before it reaches the virus, then we have a problem. And so this launched us into an entirely new investigation on, okay, well, what happens in the context of a membrane bioreactor if you have the organic matter interfering with your process? Now, this is known. We've known for some time that organic matter makes it so that the viruses, or you know, quenches these radicals that are produced. In particular, we mentioned these earlier in the semester, but hydroxyl radicals are very strong radicals, and they tend to work very well at destroying things, but that also means they can react too quickly. So if they're reacting with this organic matter stuff, then we're not happy because the viruses are not destroyed. Okay, so that leads us to this project where we had several different types of organic matter from a real membrane bioreactor treatment plant. And the types of organic matter we used were first from the filtration itself. So we had an activated sludge system, the side stream membrane, and then we took samples at 3 different times. First was right after an intense chemical cleaning. I mentioned the cleaning for the membrane fouling earlier. Typically, we just clean with physical, like shaking, agitation, to knock off the loose particles and to try to keep the membranes clean enough. But every maybe 6 months or something, this treatment plant would take the membranes out and would wash them with a really intense, like an acid bath, and then a base solution, and then maybe chlorine, and then rinsing, maybe vinegar, something like that. They really took an intensive chemical cleaning process, which they do every 6 months or something. And so what that does is it should remove the resistance. You know, we talked about membrane resistance, the R, right? The R total was equal to the resistance of the membrane itself plus the resistance of the fouling layer plus resistance of a cake layer. Well, the physical cleaning would usually get rid of the cake layer. The chemical cleaning hopefully would get rid of the fouling that's happening inside the membranes, like inside the pores and all of that. So then you're just returning to the membrane, the inherent membrane resistance by itself, because that's cheaper to operate. You don't have to have as much pressure. That's kind of the design parameter. So anyway, the clean one, we expect to have the largest pore size because there's no longer any fouling to shrink the pores. Then we had a membrane that was kind of in the middle of routine operation. It was about halfway along the cycle, in the 6-month cycle before chemical cleaning. So this was kind of a standard option or a medium case in terms of the extent of fouling. And then we had a membrane that was very fouled, almost, there was just before they took it off to clean it. And it was actually essentially about a week before it became the clean one, right? They cleaned it and then we took samples afterwards. So this was very small pore sizes. Very fouled, and that means it's actually removing more organic matter, or perhaps a different type of organic matter than the clean membrane. So that's one set of samples, was just how clean or how dirty the water was based on how fouled the membrane was. There's another set of samples that we took, and one of them is crossed off. You can't really see it because I'm in the way, but even if you could, it's crossed out because I'll explain that in a moment. So this set of samples we actually took from the activated sludge system before filtration, and then we filtered it with our own filtration setup with a 50 micrometer and then a 2 micrometer filter. We passed that through an ion exchange system so that we get rid of a lot of the cations and only have sodium, which is a little easier to deal with. Then we did a reverse osmosis to concentrate it. So we have lots, highly concentrated organic matter, lots and lots of organics, but not really many particles, right? Not much that's above 2 micrometers. So here we're concentrating the system, we're not filtering it, right? We're letting the clean water go and go away, and we're keeping that dirty part because we're interested in understanding what kind of effect the organic matter has on our systems. So from there, we then pass it through dialysis, which is kind of another form of filtration. It's like a bag filter, but you also dissolve like the silica particles with some acid, and so you're getting rid of some of the solids that are not organic in nature, and then you're keeping the type of some fraction of the organics, the fraction that are sort of physically sized as colloids. The rest of the solution goes through, we still have lots of organics, and then we pass it through 2 different ion exchange resins that have different qualities of stickiness. And so this one here, XAD8, gave us a fraction of organic matter that's particularly hydrophobic. It does not like water, it would rather stick to something that's not very charged. And then the next resin was kind of somewhere between that and hydrophilic. So this was our transphilic fraction. And then the last one, the hydrophilic fraction, the rest of the organic matter that really did like to interact with water and had maybe some charges. We weren't able to use that or keep that because there was just too much salt. And so as salty as that may have made us to not be able to study it, it just was not reasonable to separate that from the salt. And so essentially what we're left with is 3 samples: colloids, TPI, and HBO for hydrophobic. So those are the 3 fractions of DOM and the 3 membrane samples that we decided to test against the photocatalysts. That way we can see whether or not we can control the system and say that we can remove the right kinds of DOM so that we can use effective photocatalysis. So to summarize, we have a clean membrane, a routine operation membrane, and a fouled membrane. And then we have the three fractions from that bulk, which would be colloids, hydrophobic, and transphilic. Then we set out to understand something about these samples. And we used this cool imaging technique called 3D EEM, it's three-dimensional excitation emission spectroscopy. So it's essentially fluorescence. We excite the molecules with some light, and they emit some light that is a less intense, less energetic wavelength. So we can excite on the y-axis here at like 230 nanometers, and then we will emit something like 310. Like that, this point here, that would be the a molecule that, or a part of a molecule that is fluorescing when excited by 220, 230, and emits at like 310. So you can map, for all these complex organics, you can map kind of this heat map where lots of things are fluorescing right around here at around 400 nanometers when excited by 250 nanometer light. This gives us a very generic, a fairly crude but interesting understanding of what might these chemicals, these organic molecules consist of. And there's some generic classifications where in general, molecules in this region tend to be or tend to match the same signature for aromatic proteins. So there's probably lots of aromatic compounds if our samples are showing lots of signal in that area. There's other regions, fulvic-like, that's it. Fulvic acids and fulvic type of materials will fluoresce there. We have these soluble microbial products tend to fluoresce in that region. And then humic-like, which is kind of soil-like, products tend to fluoresce in that area. So we took these and we just used it as a tool to see what our colloids and hydrophobic and transphilic fractions looked like. And so here were what we saw there. And the colloids in particular looked like they were very much in the range of aromatic proteins. Maybe HBO had some of that, but more of that was in this third region, and the TPI transphilic fraction was similar. We also took this and looked at what happened before and after we applied the membranes. So whether it was the clean or the fouled or the regular membrane, and we took a look to see essentially what their signatures were like. One thing we noticed was the colloids seem to match here with, in terms of what was removed by the fouled membrane. And that actually we're showing that here. So essentially we have the sample of organic matter before it was filtered. So we have the activated sludge chamber. Then we have the membrane. And then we have the membrane effluent. So this here would be the bulk supernatant, the BS you see there. And then the permeate here, the P, is here. And then what we did with the third panel here, so we have the bulk supernatant for all three of these. And this was the fouled on the left, fouled membrane, washed, and the kind of the medium case. And so we have the bulk supernatant, then the permeate, and then we subtracted one from the other as an estimate of what was removed. Okay, so let me say permeate here, bulk supernatant here. And then this R essentially was an estimate of what the membrane removed. Okay, so we see that the fouled membrane removed a lot of these aromatic type proteins or molecules that resemble them. The washed membrane may have removed a fair bit of that too, but not nearly as much. And the generic one just sort of removed stuff, and that wasn't a big change. So then we took a look at how effective our photocatalysis was in reference to that. Now, one thing that happens if you put dissolved organics, this is a good example here, tert-butanol. When you're trying to degrade some target, and over here we have a target, this is something that acts like a pharmaceutical. It's PCBA, parachlorobenzoic acid. So it's something that maybe we want to destroy in wastewater, maybe a pharmaceutical compound that we want to eliminate when we're treating our wastewater. Perhaps we want to reuse our wastewater, and we don't like the fact that there happens to be caffeine and heroin and who knows what else in our wastewater. It just happens. And then this stuff is going to act like our dissolved organics. This is going to function that way. So when we add more dissolved organics, we see the rate at which our target is being destroyed. That's what the plot is showing. The rate of destruction of PCBA as a natural log plot. It's being reduced over time, and we see that when we have photocatalysis and no DOM, we have the steepest curve, the steepest kinetics. Now, the ultraviolet light alone can destroy some of this target. That's this case. So we have two boundaries. One, when the photocatalyst is working as well as it can, and the second, when it's just the ultraviolet light doing the work. In between, we have adding more or less amounts of the dissolved organics. And what we end up seeing is as we add more DOM, we see the kinetics slow down, so that we have that we're essentially competing for the hydroxyl radicals that are supposed to be reacting with our molecule. They happen to be competing in reactions with the tert-butanol. So that's kind of the simplified way of looking at what's happening in the organic matter. And so we applied this to all sorts of different organic matter fractions, the ones that we talked about. So we have colloids, we have TPI and HBO, and we also added humic acid, which we could buy from the store. So when we look at how they change, how the rate at which we're destroying this changes over time, or sorry, not over time, over the concentration of dissolved organics, we can actually see some interesting things. Now, this previous graph, you've seen stuff like this. This is the natural log ln of n over n naught. Now, this should be connecting in your mind with some things we've learned in the class by time. So if we show this in a y equals mx type of scenario, then we have natural log of n over n naught as our y equals m, and this we'll just go ahead and put n will be negative, right? M times time. So you should have seen this, right? This is first order decay kinetics, where we have ln of c over c naught equals minus kt, or we can say, in this case, ln of PCBA over PCBA naught equals some constant, which we can find with a linear regression, times time. So that's exactly what we're doing here. And then we've taken those linear regressions for all these different plots and said that here, this value, this is our value for K. Okay, so that's our K in per minutes. So we took this one in minutes, found K, given our data, by using a line of best fit, and that was our solution. And that gives us K for these different cases. So the higher the K, the faster the degradation's happening, which means our process is working very well. Okay. Now, when we see then that our rate constant drops when we add more dissolved organics. So we're adding more dissolved organics here, DOM, dissolved organic matter. One interesting thing what we can see is that some of them look like they're about linear, maybe, whereas, and perhaps even some have a little bit of a boost here towards the end, where it may actually help compared to one concentration or the next. But some of them look a little more exponential in their relationship, like that. And it turns out there was a study that showed this actually makes a lot of sense in terms of a mass balance where you look at the transfer of radicals in bulk solution. So just diffusion of radicals from a TiO2 particle where it's made out into solution to react with your target or with maybe some unwanted dissolved organics. And you can compare that to surface phase quenching. So if your molecule sticks onto the surface of your TiO2, your photocatalyst, then it has immediate access to the radicals. So that's one way we have pretty good rates of degradation. However, if your colloids or your humic acid, these guys, if they stick onto the surface, then you're essentially looking at an adsorption isotherm or an adsorption process where your organics are sticking to the surface and competing. And that is what causes a sharp decrease in the effectiveness. And so the goal here was to understand, okay, which types of organics are really sticking to the surface and causing a problem, because maybe we can deal with them and design a system so that they are less of a problem, or we can control the problem and maybe only let this type of organics through our membrane. So what we're learning here is some of these types of organics certainly are more of a problem than others. We also took a look at how this worked with fouled membranes versus that mid-cycle versus the washed membrane. And sure enough, what we see, and this is not even a linear plot, the zero should actually be much closer here. It should be, the zero mark should be right here if we're doing the scale, the scale is not linear, right? This is just a histogram. If we were to plot it on a linear scale, you would see that this would be very close, and then there would be a smaller gap here between these two, and then these two are linear in relation to each other. But at any rate, what you see is that same profile where some of them drop really quickly, and the others were kind of more linear. And the ones that were linear, or let's see, we'll start with the one that dropped quickly. The one that dropped quickly was the washed membrane. So this one ended up letting organic matter out through the membrane that really caused a problem for the TiO2, for the photocatalyst. And by the way, consider that this is for the same amount of organic matter. So you might think to yourself, oh, it's letting more junk through because it's cleaned. Well, that's not exactly the case. It's actually letting a different type of junk through. And it happens that that type of junk is really detrimental to the photocatalyst. So maybe if you want to use this type of operation, maybe you should keep your membranes a little bit foul, or design your membranes in such a way where it is getting rid of this stuff, even when it's perfectly clean. So that was kind of an interesting finding there. Some of these other portions didn't seem to have too bad of an impact. So for the fouled membrane in particular, even though it's letting out a lot of organic matter, that really didn't have too much of an impact in some cases. So there's an important thing here where we see that the type of organic matter, again, can be controlled with essentially by the membrane operation, and it does have a big impact. It's very important for the system. So just putting all these together in one plot here, just kind of showing those all together. We just talked about them all, but this was, if we wanted to compare the washed membrane versus the colloids, that would be these lines here, and we see that the washed membrane was actually worse than the colloids, and the colloids were pretty bad. The washed membrane was also worse than the humic acid here. So at the end of the day, membrane bioreactors may be pretty useful for using something like titanium dioxide or some other photocatalyst, especially if you already have the processes in place. I think that's key for the future of photocatalysis. There's not very many photocatalytic operations in existence because it's not very practical in most cases. So in order to find practicality, I think you really have to look towards designing the right pretreatment, and a membrane bioreactor is a great example because the bioreactor itself is not really getting rid of all the viruses, first of all, and it's also not really getting rid of the pharmaceuticals that you might want to destroy, but it does offer some capability of removing the organic matter that causes a big problem for the photocatalyst. Because we've known for a long time that one of the major issues with photocatalysis is that quenching by dissolved organics, and the fact that your system becomes a little bit useless if you have a real sample of water, a real wastewater or real natural water with the organic matter present. So the membranes with the disinfection directly after it that's a good example of where this type of technology may be able to have some application in a practical manner, which is exactly what this technology needs. It's been an interesting journey for this particular type of research because titanium dioxide in particular, doing anything, any sort of research with it is a little bit like beating a dead horse. It's been thoroughly studied, lots and lots of papers out there, and still there's almost no actual applications. So in my view, the only meaningful research is actually to identify and really study closely the problems with photocatalysts and to see how maybe we can overcome those problems. So that was the goal there. That's all I've got for you today. So I hope you found this interesting, and I hope you enjoy a few minutes extra today, the short lecture. And we will go over homework 4 in class on Thursday. We'll talk more about the exam. I think I am probably going to change the format a little bit to, and it'll be almost exactly the same experience-wise, but I might actually give you a thorough multiple choice scenario instead of just complete free response. So we'll talk more on Thursday about that. So if there are any questions, I'm happy to answer. Otherwise, I'll let you go. That way I can get back to grading your homeworks. All right, well, I will talk to you guys on Thursday.